Many of the DNA structures shown in this exercise are meant to serve as models to aid in the understanding of twist and writhe. They do not represent actual structures found in living cells. Each of the chromosomes of eukaryotes is comprised of a single linear DNA molecule. Because their ends are free, linear DNA molecules can freely rotate to change the number of times the two chains of the double helix coil about one another. However, DNA molecules can also be circular. Examples of circular DNA molecules include most bacterial chromosomes, some viral chromosomes, and small bacterial genetic elements called plasmids. For such covalently closed circular DNA, the absolute number of times the chains coil about one another cannot change. These DNA molecules are said to be topologically constrained. Even the linear DNA molecules of eukaryotic chromosomes can be topologically constrained because of their interactions with other cellular components. Since each of the two strands of cccDNA are covalently linked circular chains, and because they coil around one another, the strands cannot be separated from each other without breaking a covalent bond and opening the circle of one of the strands. The two strands could be separated if they were able to magically pass through each other. The number of times one strand would have to be passed through the other strand in order for the two strands to be entirely separated from each other is called the linking number. The linking number is always an integer. The linking number is an invariant topological property of cccDNA. No matter how much a cccDNA molecule is distorted, the linking number remains constant. The linking number is the sum of two geometric components called twist and writhe. Twist is simply the number of helical turns of one strand about the other. In other words, the twist number is equal to the number of times one strand crosses in front of the other strand. If cccDNA is lying flat in a plane, the linking number is equal to the twisting number. If the twists form a right-handed helix, as in DNA under normal conditions, they are given a positive twist number. To determine if a helix is right-handed, think of it as a screw. You would have to twist a right-handed screw to the right, clockwise, to screw it into a board. Also, in a right-handed helix, the strand in front If the twists form a left-handed helix, as can occur under special circumstances, they are given a negative twist number. You would have to twist a left-handed screw to the left, counterclockwise, to screw it into a board. Also, in a left-handed helix, the strand in front at a cross point is pointed up to the left. However, cccDNA does not generally lie in a flat plane. Instead, the DNA double helix usually crosses over itself in three-dimensional space, often repeatedly. This torsional distortion is called writhe. Writhe can take two forms. One form is interwound or plectonemic writhe. In interwound writhe, the DNA is wound around itself, forming a helix. Most cccDNA molecules exhibit interwound writhe. The writhing number is the total number of writhes in a molecule of DNA. If right-handed DNA forms a right-handed interwound helix, the DNA must twist to accommodate the writhe. Therefore, right-handed interwound turns are assigned negative writhing numbers. Note that, although the linking number must be an integer, the twist and writhing numbers can have fractional values. If right-handed DNA writhes to form a left-handed interwound helix, the twist number of the DNA decreases. Therefore, left-handed interwound turns are assigned positive writhing numbers. The other form of writhe is spiral or toroidal writhe. In spiral writhe, the DNA winds as if around a cylinder. Spiral writhe often occurs when DNA molecules wrap around proteins. If right-handed DNA writhes to form a right-handed spiral coil, the twist number decreases. Therefore, right-handed spiral turns are assigned positive writhing numbers. Note that this is the opposite of right-handed interwound turns. If right-handed DNA writhes to form a left-handed spiral coil, the DNA must twist to accommodate the writhe. Therefore, left-handed spiral turns are assigned negative writhing numbers. Again, this is the opposite of left-handed interwound turns. Under normal physiological conditions, the average structure of DNA has about 10.5 base pairs per turn of the helix. Therefore, the linking number of fully relaxed DNA can be calculated by dividing the total number of base pairs by 10.5 to find the number of twists. This linking number is assigned the symbol LK0. If writhes are introduced to the DNA without changing the number of twists, the linking number changes. The difference between the actual linking number and LK0 is the linking difference. If the linking difference is significantly less than 0, the DNA is negatively supercoiled. If the linking difference is significantly greater than 0, the DNA is positively supercoiled. cccDNA from both bacteria and eukaryotes are usually negatively supercoiled. Negative supercoils can be thought of as a store of free energy that aids in strand separation for processes such as DNA replication and transcription. When right-handed interwound turns in the DNA are relaxed, the twist number decreases and the DNA unwinds. Ask yourself, under what conditions might an organism need positively supercoiled DNA? DNA molecules with the same sequence and length but different linking numbers are called DNA topoisomers, since they differ only in topology. Topoisomerases are enzymes that change the linking number of a DNA molecule. There are two types of topoisomerases. Type 1 topoisomerases increase linking numbers in steps of 1. By introducing an additional twist, type 1 topoisomerases relax negative supercoils. Type 2 topoisomerases change linking numbers in steps of 2. Type 2 topoisomerases require energy from ATP hydrolysis for their action. By changing the handedness of turns, type 2 topoisomerases change the topology of DNA molecules. Prokaryotes have a special type 2 topoisomerase called DNA gyrase. DNA gyrase decreases linking numbers in steps of 2. DNA gyrase is responsible for the negative supercoiling of chromosomes in prokaryotes. You have discovered a protein with an amino acid sequence similar to known topoisomerases. How can you determine if your protein is a topoisomerase? The greater the writhe of a topoisomer, the more compact the shape of the cccDNA. Question 1. Of the techniques listed, which could you use to differentiate between supercoiled topoisomers and relaxed topoisomers? Correct. DNA affects its functioning in the cell. Cells use the torsional strain of negative supercoiling to promote strand separation for processes such as DNA replication and RNA transcription. However, if DNA becomes too negatively supercoiled, the strands will separate too often, making the DNA more susceptible to degradation. The topological state of DNA is controlled by a class of enzymes called topoisomerases. Topoisomerases are divided into two broad classes, type 1 topoisomerases and type 2 topoisomerases. When you have completed this exercise, you should understand how many type 1 topoisomerases can increase the linking number of a DNA molecule by cleaving one strand of the DNA, passing the other strand through the nick, and ligating the nicked strand. Note, this animation provides an example of the reaction pathway used by many, but not all, type 1 topoisomerases. These type 1 topoisomerases can only bind to segments of DNA in which the two strands are melted, that is, segments where the strands have separated. One of the DNA strands binds in a cleft in the type 1 topoisomerase, placing it near the active site tyrosine. The active site tyrosine becomes deprotonated, probably by a nearby basic amino acid residue. The result is a negatively charged oxyanion. The oxyanion attacks the phosphate group at the 3' position of the nearest deoxyribose, producing a DNA 3' phosphotyrosyl enzyme intermediate and a 5' hydroxyl polynucleotide. The hydrogen atom on the 5' hydroxyl is probably donated by a nearby acidic amino acid residue. After cleavage, the enzyme undergoes a large conformational change. This conformational change opens up a gap in the cleaved strand, with the enzyme bridging the gap. The uncut strand then passes through the gap, binding to a pocket in the middle of the enzyme. Binding this strand causes the topoisomerase to change conformation again, closing the gap in the nicked strand. The topoisomerase then ligates the nicked strand by reversing the cleavage reaction. Finally, the topoisomerase undergoes another conformational change, opening up and releasing the DNA. Overall, the reactions catalyzed by the type 1 topoisomerase have added one twist to the DNA, increasing the linking number by 1.